Okay. All right, thank you, everyone, and thanks to the organizers very much, because it's not very often that we manage to take part to a symposium that is exactly specialized on this type of topic. So it was an amazing opportunity. What I will be presenting today is the outcome of two studies that have lasted 4 years, the length of my PhD, which I've done at the University of St Andrews. And they focus on Temnothorax nest wall building. I'm an evolutionary biologist, so the first of these studies will be actually on an update on the current behavioral model of Temnothorax, how they build the nest wall. But the second part will focus on a question that is of interest to me, which is the transition between one collective phenotype to another. So how does the system preserve the robustness and the quality of the collective phenotype when different individuals are doing different things? We're talking about mutations, which are the key ingredients of evolution. All right, so let's talk a little bit about the nesting behavior. The characteristic of the genus Temnothorax is that most species are very small, and many of them nest within rock crevices. As you can see in this picture. In particular, I will be focusing on two species that are known to build a wall within the crevice that they inhabit, Temnothorax albipennis and Temnothorax rugatulus. If you translate any of these two species within a lab and you allow them to build freely within 2 microscope slides, you will see that the structures actually is circular. So you can approximate it with some mathematical rules, and importantly, you can approximate it as a 2-dimensional system, which of course makes computational modeling much easier. The function of the wall is not entirely not known so far, but we think that the main one of the main functions is defense. So for this reason, we can assume that a functional wall must have 2 features. The first one is we don't want any gaps, and the second one is that it must be quite compact to resist any external pressure. All right, so I will start by talking about the behavioral model. And most of you will probably be familiar with the original model, which is the one proposed by Funks and Denneberg in the 90s. This model is made of 2 components. One is the template, which is believed, there is actually no empirical evidence for this, but it's believed to center around the root cluster. So this, the optimal distance, built-in distance, or the optimal location at which to build depends from the distance of the location from the root cluster, which is kind of proportional to colony size. At this optimal location, you have a positive feedback on deposition and a negative feedback on pickup. And the farther away you are from this location, the less likely deposition is, and the more likely the removal of material is. The second component is stigmergy, a stigmergic effect, which is exercised by the amount of deposited stones, so the size of stone piles. And again, in the original model, there is a positive feedback on deposition. So the larger the stone pile, the more likely it is to grow, and a negative feedback on pickup, so stones are less likely to be removed. There are a couple of problems with this original model. The first one is that if there is insufficient building material, then the resulting wall is poor quality. The second problem is that this is a model of the beginning of building behavior, so there is a lack of termination. What I have found is that there is at least one more component to the model, and particularly in the stigmergic component, it is true that there is a positive feedback but it only occurs at low stone density. So when let's say the stone piles are quite small, while as the stone density increases, negative feedback kicks in, which allows for behavioral termination. And what you see in the picture at the center at the bottom are some laboratory data of Temnothorax rugatulus. And as you can see, we have this almost perfect bell curve shape. So there is a single cue that regulates the whole length of building behavior. And now that we have a model, we can move on and ask some evolutionary questions. So what I'm going to do is exploit the same type of simulations, and I'm going to ask what happens if we introduce a change in the parameter value of the template rule. So we have a quantitative mutation, and which is equivalent to a change in the optimal building distance. And I'm going to estimate the impact of these mutations, this individual variation on wall quality. We'll measure with two measures. So without going into the mathematical details of this, they are circular dispersal and distance dispersal. So the first one tells us whether there are gaps in the wall. And the second one tells us how compact is the wall. So it's essentially the dispersal of the stones as you move away from the optimal building distance as defined by the template component. And the closest these two measures are to zero, the better the wall. So let's start. I will look at three scenarios, and I will start with the first scenario, which is the one that is more clearly evolutionary, which is what happens in the case of dependent colony foundation. So in this case, we have queen adoption or readoption, and what happens if the new queen carries a mutation in the germline, or the workers that it produces carry a mutation, maybe even from the paternal line. When the old queen dies, then we have a slow transition of the worker population from the original variant to the new variant. And what I want to know is what happens in the middle when you have individuals who use different variants building at the same time. All right, so on the x-axis here of this figure, we have the difference between the resident and the mutant distance rule, which is also a measure of the conflict between these two variants, because of course, the more far apart the two optimal building distances that these two variants define for the worker, the more conflict there will be. On the x-axis on the left, we have our measure of how compact the wall is, and on the left, and on the right, there is our measure of how many gaps there are in the wall. And each panel is a different frequency of the mutant in the population: 10%, 30%, and 50%. So 50% each variant is equally represented in the population. And I'm also comparing two models. One is a full model in blue. In red, which has both the template and the sigmoid component, and the other one is the template component only, because what I'm aiming for is to break down the role of different components of the algorithm on robustness. So there are three things that I want you to pay attention on, and the first one is that overall the collective phenotype, the quality of the wall remains good. And it will become more obvious in a second when I show you. The second thing is that if you look at the circular dispersal, so how circular the shape is and also how many gaps there are, it's almost fully unchanged no matter the conflict and no matter the frequency of the variant. And the third one is that the wall, if we take out the stigmerging component, the wall loses its compactness. If we look at the figures, this is much clearer. So almost in all cases, we have a fully functional wall, which is compact and circular, and there are no gaps. I've also looked at two different types of variation in the colony. The first one is the presence of multiple variants, a few, 4 in this case, which can be approximated as the existence of multiple parental lines in the population. This doesn't happen in Temnothorax, so I'm using this particular model as a study system. And on the right, on the other hand, you can see what happens if there are as many variants as the number of workers in the population. So we can think of this as epigenetic variation. And as you can see, I didn't have space to show you the pictures, but the quality is still very good. So the collective phenotype is robust to the presence of this variation. I've looked also at different relative frequencies of the parental line. And the only case where we have some outliers where the wall is really bad is when if we allowed for huge differences between different variants. So that's the case on the right where the standard deviation is 5. And again, we see the same other two effects that I mentioned before. So there are no gaps in the walls, almost in any case. And also, if we take out the sigmoidic component, then the wall, the stones are more dispersed and the wall loses compactness. What does it tell us in terms of the role of the different components of these algorithms? My hypothesis is that there are two components that are key for this effect. The first one is the local activation long-range inhibition mechanism, which in this case is stigmatry, which drives the convergence of multiple variants. And that should not surprise us because that's the same mechanism that drives collective decision-making and the conversion to one solution. And building behavior is a form of collective decision-making. And the second one is that there must be an area of overlap where building occurs under all rule variants with high probability. And I've called this buffer zone. And what happens, I've looked at what happens when you reduce, in my simulation model, the size of the overlap. And as you can see, we start seeing the formation of two different walls. One is like the other, because you reduce the area where the convergence component, the sigmoidic component, can be put in NACT and drive the combinations of the two components. And so, and you see accordingly that if you take out the sigmoidic component, the two walls are completely separated while they're still trying to merge if you keep the sigmoidic one. So what does this mean? It means that we expect, at least this algorithm, to be very robust to the presence of mutations and to the generally to individual variation. So we can also start thinking of what this means when we are considering the evolution of these building algorithms in relation to, for example, the evolution of dependent colony foundations and other sources of colony variation within the colony. And second one is the second thing to think about is that the size of the buffer zone probably plays an interesting role in building behavior in general. So not just evolutionarily, but also when we think of what is happening when we move from the use of one, from building one structure to the next one. Okay, thank you.
Thank you, Edith, for this very interesting talk. So I'm very interested to read your forthcoming paper.